Hi friends. As I said in the previous revision class, today we are starting methods in molecular biology. So I have divided this into three parts for your convenience. And today we will go with the very basic methods. All these molecular methods starts with any of the isolation protocol, just like DNA, the Delaporte or CETAP method, RNA, the trisol method, or protein with any phosphate-based, phosphate buffer-based isolation protocols. So normally we do this in our laboratories for our all plant-based molecular biotechnology technique. What we do is, we do start with any of the basic material, whether it is a plant sample directly collected from the field or from glasshouse, or it is grown in growth chambers, or some of the cell cultures, what we prefer having this vectors and other things multiplied in that. So this starting material should be structured to break open the cells to release its contents in the buffer so that further it can be purified. So this is the normal protocol for any of these isolation procedure. And in case if it is based on some of the cell cultures what we are using in the protocols, we go for testing the sufficient enough cells presence in the broth. First, this is being tested by a spec having 600 nanometer wavelength. And normally we fill this cuvette with sufficient enough broth culture, and the OD is recorded. So normally this formula is used actually. So the OD unit is equal to the intensity transmitted by intensity incident. So based on this, we calculate the OD value, and one OD unit is actually approximately equal to 0.8 into 10 power 9 cells per ml. So this much is required to go for further procedure. So after this, the cells are being harvested from the bacterial culture just by centrifugation, and then the supernatant broth is removed, and this pellet is resuspended to go for extraction, and resuspension is normally done in a buffer. Okay, so many times this buffer contains one detergent. This detergent will be having the lysis action, just like the CTAB or SDS. So these two are normally detergent used in buffers to break the cells to leave the cell contents in the buffer. And second one, the pH of the buffer. The pH of the buffer depends on what kind of material you are going to isolate. Normally, in DNA isolation protocols, our pH of the buffer is a little bit high. It will be in alkaline range around 8. Okay, 8 or 8 plus it is. So for RNA and protein, it differs on the purpose how you are doing and using which buffer you are isolating. And this isolation buffer contains salts to maintain the pH of the buffer, as well as some of the ions to chelate the contents in a different stage. Okay, so that is very basic components of any of the buffer what we are using. And after this preparation of extract, cell extract, we go for phenol-based separation technique. Normally, it is phenol chloroform isoamyl alcohol used in the range of 25, 24, and 1%. So many times it is only the phenol and chloroform. And this mix normally used to actually coagulate the protein present in the cell extract. And just by that, the phenol as well as the coagulated protein layer is separated from the actual aqueous mix, which is having DNA, RNA as well as protein present sometimes, because all proteins are not precipitated in the single step. So sometimes there can be proteins also present in the aqueous stage. Okay, many times it is only DNA and RNA, but there can be protein also. So if there is protein contamination or RNA contamination while you are isolating DNA, then you have to go for protease and RNase treatment to make them further purified. Otherwise, you can go for very specific column-based purification technique, whether it can be either it can be an ion exchange chromatography or column chromatography, which is based on silica particles. In case if it is ion exchange chromatography, normally the salt is replacing the DNA molecule. So just by that, first it is bound to that column, and after that it is extracted by adding more salt. Whereas in silica particles, it is first based on water only. So this water, or it is added with some of the minerals, means like it is kind of a low concentration buffer is used to extract these DNA molecule. Okay. So all these things are based on the standardized protocol for each of the sample we are using. The next one is purification of plasmid. This is a little bit tricky. Many times it is being done using cesium chloride gradient centrifugation. And while though this procedure looks very simple for reading, actually this is very complicated kind of handling these kind of cesium chloride gradient centrifugation and adding Ethereum bromide to it and separating the particular band as well as purifying that with an butanol. Okay, so this butanol step normally removes this Ethereum bromide in an organic phase, whereas this DNA is left in the aqueous phase. At last, this DNA is to be dialyzed to remove the cesium chloride using a calcium chloride diffusion buffer. Okay, so this method is a little bit tricky and it needs expertise and like hands-on experience is needed for this purpose. Apart from that, many times we use enzymes in our manipulation techniques. Some of these are exonucleases. Exonucleases can cleave at both the end, both the strands also. Otherwise, it can be from only one strand, like only from 3' end or 5' end. So it depends on the enzyme it acts. And there are endonucleases. There are 3 types of endonucleases. Restriction enzymes you might have already studied. And type 1 and type 3 are not that much specific when comparing the type 2 restriction enzymes, just because of its specificity in recognizing the restriction site as well as its breaking site, that is the cleavage site. Type 2 enzymes are preferred in gene manipulation. So next thing what we have to learn is PCR. The PCR is a very basic technique in any of your laboratory procedure. And we have already isolated the DNA, and that DNA can be used as a template DNA. Before running PCR, first we should go for checking the quality as well as quantity of the DNA using SPEC as well as gel electrophoresis techniques. You can run and check whether there is intact DNA or there is protein or RNA contamination present in the DNA. So after all these steps, you will be diluting the DNA and making a PCR mix with the DNTPs and polymerase enzyme, and then buffer, everything will be added to that. And this PCR mix is loaded to the PCR machine, and you will be programming it for a particular number of cycles. Then the primer concentration, that is much more important. You have to calculate exactly the required primer concentration, and you have to add the primer sufficient enough for the reaction to go on. And in the first step is normally a denaturation step. It happens at 90 plus temperature, and this is also standardized in many cases. It should be standardized for how many seconds it should run. Okay, so normally we write like 94 degrees centigrade for denaturation. And the very next step is annealing temperature, and this is based on your TM value. Please read about TM value once again. how it is calculated and the formula for calculating that also you should know. So just go through that once. And after this annealing, what happens is primer extension happens. That is synthesis of the new strand happens. And this happens in 72 plus temperature. And this cycle is repeated for many times. After 25 to 30 cycles, at last everything will be done. And the final extension you will be giving for 1 or 2 minutes, and at last it will be hold for 4 degree or less than 4, whatever it is. So once the contents are cooled, and then you can remove it and check by running the electrophoresis. Okay, so that will be the next step. And normally in a PCR reaction, what happens is the primer comes and binds, and after that the newly synthesized complementary bases will be added. Means the bases added in the reaction mix is added to this newly growing chain by the polymerase enzyme added in the reaction mix. And normally DNA polymerase is having one kind of nick closing activity, and this nick closing activity is what actually it does is, if it is DNA polymerase, it adds bases to this nick, as well as it replaces the existing onward sequence of the DNA. Okay. So whereas if you use a clonofragment, it closes only the nick. It is not changing the existing nucleotides of the growing chain. Okay. Whereas in case of reverse transcriptase, it uses RNA as a template, whereas the newly synthesized is a cDNA. Okay, it is a complementary cDNA. And next one is gel electrophoresis. Whatever you have done, whether it is a DNA isolation or a PCR or a restriction reaction, then you will be going for a this one, electrophoresis. Normally what we use is an agarose based submergence electrophoresis. And the submergence electrophoresis normally runs from negative to positive. In the sense, you can keep that in mind, like the negativity in your mind should always run towards searching a positivity in your life to succeed in life. So just keep this quote in your mind so that you will always remember that the DNA with a from the negative side should always run towards the positive side. Okay, so just keep that in mind while running this agarose gel. Normally the ethylene bromide runs from the positive to negative, whereas the DNA runs from negative to positive. So that once the ethylene bromide moves towards the DNA, it gets intercalated into the DNA molecule. So this intercalation we have seen in your first revision class as well as second revision class also. If you want to recollect it, you can just go through that again. Okay. Then after running this gel, you can use one transilluminator to view your gel or gel doc unit to take a picture of it. Apart from that, enzymes what we have seen earlier, normally we use alkaline phosphatase, phosphonucleotide kinase or terminal transferase kind of enzymes also in some of the procedures. Alkaline phosphatase, you know that actually it removes the phosphate group present in the 5' end. Okay, so normally we say like a 3' and 5' end of a sequence means you should remember exactly the ribose sugar. And in ribose sugar already I have shown you that in the 3' end there will be a hydroxyl end, and in the 5' there will be a phosphate end. Okay, so that you should remember. And this alkaline phosphatase removes the phosphate group present in this 5' end so that it turns into a hydroxyl end. Why this is required is when we cut any of the vectors using any restriction enzyme, it will be having this active phosphate groups. And this active phosphate groups will make them again recircularized. Means the vector will go for closing it again. So to prevent this recircularization of the restricted vectors, we use this alkaline phosphatase. Normally, we use the off-intestine phosphatase that is known as SIP. Okay. So next one is polynucleotide kinase. This polynucleotide kinase acts just opposite to the alkaline phosphatase. If we are having anything with only hydroxyl groups and 5' and also there to add phosphate groups, this phosphate polynucleotide kinase is used. Then one more enzyme is terminal deoxynucleotide transferase. This enzyme adds either one or two nucleotides at the ends of these growing chain. Okay, so it will be added to the 3' end in case of a single strand, and double strand it will be added in the both strands. It will be added. You can see it here. So in both end it will be added. Okay, in the 3' end. Next one is gene cloning. This is very interesting area what you asked for actually. Normally the vectors are taken from any bacteria, kind of, whether it is a modified vector or constructed vector or designed vector, whatever it is, whatever you call, it will be having a biological origin actually. Okay. So at last you are having one vector with you, okay, vector of your interest. Then you have identified a gene of interest, like whether it is directly isolated from the DNA and restricted with the restriction enzyme, or you have amplified it using PCR with a known primer and then isolated from that amplified product by gel elution technique or whatever it is, some kind of pure fragment you are having. Okay, so both this quality and quantity, everything you know. Then after that, once you go for the ligating procedure, then at last you will get one recombinant clone, and this recombinant DNA molecule is inserted into a new bacterial cell. Okay, so that means normally we'll draw them in E. coli. So this recombinant molecule is added to this, and at last you will be growing this recombinant molecule carrying bacteria. Many times this recombinant DNA molecule will be in multiple copies. At last you multiply this and then go for screening of this, means selection medium. So if these vector is having resistance to anything like Basta or ampicillin or anything else, okay, whatever gene is present in that, it will be having resistance towards it. And using that selection medium, you can plate it or you can throw in broth also and select the recombinant molecule. Okay. So this one is explaining the same thing. Actually, here they have used RP4 vector. RP4 is a resistance contributing vector. So all these things I am showing directly from your textbooks, recommended textbooks. You just go through it again. So for your easy remembrance only, I am showing directly your textbook. Okay. So next one is, so these vectors are having so many resistant genes, and once you clone it, you will be having a lot of mix of recombinant versions as well as non-transformed ones also you will be having, means the vectors with the recombinants. Okay, so recombinants will be present, or otherwise the cells without the plasmid also will be present. So to separate all these things, no, we are interested in the recombinant plasmids. So what we will do is we will have the growth medium with the selection antibiotics. Okay, so just by adding these tetracycline or kanamycin, ampicillin, whatever it is, so just by that we prevent the growth of cells without the resistance contributing plasmid. So just by that we can pick it and then go for separating the plasmids again. Now we do have different kinds of plasmids. The one is non-integrative plasmids. These plasmids will remain separate from the bacterial chromosome, whereas we do have the integrative plasmids, and these integrative plasmids will form episomes. Whereas if it is bacteriophage, it is known as prophage also. Like then in case of conjugal type, normally it goes through this pilus. Pilus are first connected between these donor as well as the recipient cell. At last that moves to the negative cell from the positive cell and forms a copy of it. Okay. The bacterial plasmids are of 5 types, the basic classification. No need to remember in the same order or whatever it is, just know what it is. So that is more than sufficient. The F plasmids are actually having a tragene or transfer gene kind of thing. That purpose of this plasmid is not that much explained. That is, the function is not exclusively elucidated yet, but they know that this kind of transfer is happening. That's all. The next one is R plasmids. Just now we have seen RP4, and this RP4 is identified from Pseudomonas. Next is col plasmids. Col plasmids code for colchicin, colicins. These colicins are actually toxic compounds for other bacteria. So whenever plasmids containing this col plasmids are present in any bacterial cell, and this bacterial cell will produce a kind of toxin which prevents the growth of other bacteria in that medium or in that environment. Then very next type is a degradative plasmids, and these kind of plasmids are identified in Pseudomonas putida kind of organisms, and they do have degradation properties of particular compounds like toluenes and other things. And where plasmids, it is all well known to you. The best example is TA plasmids present in Agrobacterium tumefaciens. And regarding this bacteriophages, already we have seen about these lytic as well as lysogenic cycles in our previous discussion sessions. So I'm not going into much detail of this lytic and lysogenic cycle, but you just remember that it will be having a hexagonal head and a tube and legs, 6 legs kind of structure will be there. It goes and attaches to the surface of the bacterial wall, and then it injects its linear DNA into that. And just by that, the DNA molecule of the bacteriophage enters inside the bacteria, and at last this starts multiplying using the host machinery. And whether it forms prophage or it goes for lytic cycle, it depends on the activation of presence of the lysis kind of gene. Means there is a specific region of the genome which is actually regulating the lytic or lysogenic cycle of the bacteriophage. Okay. Whereas there are some other kind of vectors, means phages. M13 normally, this is also used as a vector, major vector, means many times we use this one. And this actually have a different kind of cycle actually. Okay, so that we will see in the next coming slides. This image actually represents a lambda genetic map. This is actually a linear one, but just because of presence of 12 base sticky end at the both the ends, it forms a circular molecule. Okay, and once it starts replicating, it replicates like a catenin, and then this one actually follows a rolling circle kind of model, and at last it produces continuously long thread of double-stranded DNA kind of thing. Means it produces long stretch of DNA, and at last at its cast site, it's being cleaved to form a separate molecule, and at last everything is packed to release the virus particle. This is all about phage DNA, lambda DNA. Whereas M13 enters through the pilus. Okay, so M13 attaches to the pilus, and it sends its genetic material inside the bacteria, and M13 replicates inside the machinery, and virus shedding happens simultaneously without even breaking its host cell. Thank you. We stop here.